Good morning, everyone. We are going to be presenting our project, which is pacemaker for biomedically engineered cardiothread e-suture. Our presenters are myself, Bryan Ceron-Espinosa, Rhianna Lee, and Melvin Soriano.
Okay, our talk today presents a 7-year effort to address a gap in potential therapies and biomedical devices for the loss of cardiomyocytes after cardiac injury. Our lab has designed experimental approaches that incorporate human cell culture applications, regenerative medicine, and electrical pacemaker design in order to target several cardiac issues. There's a great need for cardiac disease therapies, and cardiac disease is currently the #1 cause of mortality in adults worldwide. Some research efforts focus on replacing the loss or dysfunction of cardiac muscle cells and increasing pump function, which are common deficiencies caused by cardiac disease. Numerous conditions, including genetic mutations, can cause deficiencies, and we believe our current work described in this presentation may potentially address some of those. Students are provided with P19CL6 mouse stem cells, 1% DMSO, and 1, 5, and 10% complete media, depending on the desired cell growth, as well as chromic gut sutures, thrombin, fibrinogen, and adherent cell culture plates. These materials were chosen based on established literature showcasing the CL6 line as being the most useful to study in vitro cardiac muscle cell differentiation. These same cells are used to grow myocardial tissue on fibrin matrices. Replacement of fibrin with cells, as in our experiments, mimics the body's own natural repair processes in replacing damaged tissue. Cells are then monitored using inverted microscopes, digital cameras, HD monitors, and imaging software. Some important results from earlier experiments include beating cardiac cells within fibrin structures. Here's one example.
Okay, this is a structure that we found.
So this is a structure, we nicknamed it gear, and it is within a DMSO gelatin well. This is another example, we nicknamed iguana, which has numerous beating centers.
Okay, this is Ivan's iguana. We're going to go.
And then our third example, which is the most useful in terms of this project, is nicknamed funnel cake, which depicts pacemaker tissue tendencies. Our other finding was from the summers of 2016 and 2017, where we were able to 3D print the shapes that led to a suture thread embedded with precardiac cells. The idea behind this banjo project was that these sutures seeded with P19CL6 cells could be sewn into areas of the heart that have reduced contractile function and fewer cardiomyocytes due to myocardial infarction, for example.
Then from right here, you can see that our banjo project, it sort of mimics the conditions that you find in robotic-aided seno-arterial surgery. Basically, we have these suture holes in the center of our model disc, which allows for easy threading, where we combine our cells onto the suture. And then from there, we have a sort of suture which is called the chromic gut, which has translucent properties which allow us to view them and see what kind of structures our cells are able to produce. And with hydrogel, we keep everything encased in a sterile environment in which our cells can thrive and not be disturbed. This is one such example. Can you go back? From right here, you can see our suture. The material is very translucent. From there, you can see different beating centers that occur throughout the suture. And from there, you see different sites start beating either in unison or in different types of beating centers.
Our summer 2018 results are called the ring disc experiment, which built upon previous summer innovations by utilizing seated sutures, which were then used to repair porcine heart tissue, mimicking difficulties encountered during heart valve surgery. We were able to encourage cell growth both on the porcine heart and valve tissue, as well as between the sutures. This addressed whether a surgeon could potentially introduce the seated sutures into cardiac tissue during surgery, have the cells migrate from the sutures onto adjacent cardiac tissue, and then provide regeneration to the areas that have lost these important cardiomyocytes.
A ring disc, from what Rhianna just told you. We have two housing locations for different types of cell tissue. We have the porcine and then we have the murine, which is pig and the cow. From there, we're able to run our suture right through the center and interconnect both sections. And through our research, we find that they're able to create a bridge where it expands the gap, which gives us a proof of concept in which we can see that this is possible for future direction, which is most likely therapeutics. From right here, you see macrostructures start forming in the center. Beforehand, what we had was beating centers throughout the suture. Now we have giant bodies that start forming towards the center.
And our most recent 2019 project designed an e-suture frame which holds the suture hydrogel, which is made of fibrin and cells, as well as the pacemaker and Raspberry Pi.
The Raspberry Pi is a friendly user interface which is able to program our beating sutures. Basically, this project, the e-suture frame pacemaker, is an example of our research which led to creating a sort of programmable user interface which will allow us to program ourselves and have a sort of unified beating instead of what we had beforehand, which is different beating sites that were not synchronized. Now we're able to synchronize them and actually mitigate any differences in frequency. This is one of our models, which we programmed and designed in CAD. Melvin will talk about it now.
As Bryan was saying, in our 2019 experiment, the e-suture pacemaker, we had to create a new design that would allow an electric current to interact with our cardiac cell interface. And to do this, we used a program, Fusion 360. And in Fusion 360, we created these 2D sketches on various planes that we would then extrude using an extrusion tool, which would create bodies, which would create 3D bodies that we would then manipulate and cut. And from there, we would take these 3D objects and form them into a prototype that would be printed on a user-friendly printer, the Lulzbot TAS6 printing system. And the filament used for the e-suture frame is yellow PLA, which stands for polylactic acid, and the filament used for the modified lid, which we would want a more clear window to examine under a microscope, is the Tolman T glass filament. Okay. And for the modified lid, the modified lid is a replica of a standard 6-hole Petri dish lid, but with added threaded posts to hold these conductive springs. And guide rails along the sides to keep these springs from leaning to the side. We also had holes adjacent to these threaded posts that will lead outside to the pacemaker. Now, to have an electric current run from a pacemaker through this modified lid and to the cardiac cell interface on the e-suture frame, we had threaded posts also on the e-suture frame, which would hold conductive springs. These threaded posts would be labeled as positive and negative, as in a circuit. And the perpendicular alignment from the conductive springs to the cardiac cell loaded sutures would induce an alternating current within the interface.
This is our entire setup.
Device hooked up to the pacemaker on the camera.
This is our setup, the apparatus, the viewing microscope,
And here's the
our
image
display.
coming off the screen.
the Raspberry Pi, we
Raspberry
can
Pi with
program
circuitry
the duty, the frequency,
and
and then the
computer program
pseudopython,
that's running.
which is a Python shell, which allows us to program what kind of voltage that we want to deliver on our cells.
Here's the device.
So the concept of a pacemaker, we have two examples, a permanent and a temporary. What we're trying to achieve is a temporary pacemaker. We have two different locations, two different leads, which is the cathode and the anode, which in a close proximity of each other, we're able to deliver a frequency or a voltage and allow us to stimulate those areas for a unified beating area. Potential. But there is a correlation between age and myocyte autophagy. Sorry. Autophagy is when cells destroy themselves due to various stresses. Something occurs in the cell, mutations. The cell will lose a part of itself. Mitochondria, for example, and the body needs to destroy that in case it leads to something else, maybe cancer. Some of these examples are, we have a damaged organelle, a nutrient deprivation, or hypoxia. This can occur with advanced age, most likely the elderly. What we're trying to combat is once that occurs, like a myocardial infarction, we're able to repair that with the utilization of IPS and TBX18. IPS is induced pluripotent stem cells, which means what we have is a mature cell line, such as your skin. And with induction, we're able to transfer that back to an embryonic state, which then we can induce with TBX18 and change it to a myocardial-site, which then we are able to implant into the region that was affected and reform the sinoatrial node, which is basically the pacemaker cell in the heart.
Okay, so the adult heart does not efficiently generate new cardiac muscle cells in response to injury, even with temporary drugs and current mechanical devices that we have nowadays. So this demonstrates the need for innovative strategies for heart regeneration. We believe our seated structures could be used during surgery for congenital heart defects that feature holes in the septum or in the center of the heart. Septum holes occur in atrial septal defects and ventral septal defects, while complete atrioventricular canal defects leave holes in the dead center of the heart. A hole in the septum can cause blood to flow in the wrong direction or even the wrong place, while a hole in the center of the heart affects all four chambers. In these cases, our seated sutures that have been shown to grow on cardiac valve and muscle tissue, as well as bridge gaps between sutures, could potentially be of use during surgery. Our most recent pacemaker model is a simple and inexpensive model that addresses surgical wound complications, reduces infection, increases cellular immunity, and even can improve surgery results in the future. Our immediate goals in the future involve demonstrating our cardioethrid suture pacemaker model as a directional beating patch for repairing gaps in the heart tissue. As well, we would like to modify our CAD model as an electrically assisted healing device for dermal lacerations. This design is based on a paper that demonstrates an e-bandage invention that applies low alternating current to generate a magnetic current that reduces wound healing time. Additional smaller changes we'd like to have happen include replacing the silver components of the pacemaker model with platinum, because platinum is more likely to encourage cardiogenesis. We would also like to acquire 2 robotic surgery units so that students can incorporate this in technology during their suture technique. The seated sutures on porcine cardiac tissue could potentially assist in healing bisected heart valve tissue during robotically assisted, minimally invasive heart valve repair. Lastly, we would also like to expand on applications for our cardio e-thread and improve previous experiments. Our references section includes the publication of the P19CL6 differentiation paper with DMSO, as well as our lab's YouTube website and our principal investigators curriculum model. This concludes our presentation. Thank you for listening, and let us know if you have any questions.
Thank you very much. Any question? Yeah.
By the way, everybody's going to get questions from me, so you know. So you mentioned this is like a 7-year project. I'm presuming you guys haven't been a part of that project for this whole duration. Can you elaborate a little bit about your individual contributions?
Yes,
And like when you started on it?
Oh, I've been there since 2016.
Okay.
I designed the banjo and the ring models.
Yeah, and then I've been here since the ring disc and well as the pacemaker model. So I was not there for part of the banjo and then the very simple structures that they had beating in the gel.
I've been here since the previous summer, and I've been CADing the discs that we use. And currently, I CADed the e-suture frame.
Any other questions?
What are the voltage levels that you're stimulating with? You said you're creating in an alternating
Yes.
way. What are the amplitudes?
So the max output for the Raspberry Pi, which I have in my hand, is 3.3 volts. From what we're doing right now is only applying 5%, which is roughly around 1.126 voltage, only because we don't want to overstimulate ourselves. And we changed the duty cycle, so it's not that strong, but we also changed the frequency so the hertz are not too much. So we stimulate it at various peaks.
So when the suture is installed in the heart, so usually it mimics the heart application.
Oh, so what we're trying to do with future direction is that we'll grow the cells in these wells, the square wells, and from there we're going to extract them and place into the heart. What our pacemaker does is copy what the person's heart would be like, would start beating, and from there we extract it out from there and in vitro surgically in place.
What's the range like? Usually because the heart sometimes has a different.
Yes, sir, it beats in different frequencies.
Yeah. So it does adjust automatically or matches whatever it receives.
Yes, from what we've seen is that once we surgically extract it, we just beat it so that all of them start beating at the same time. We place it inside the heart. From there, we'll start matching the rest of the body.
What happens if there's a conflict or mismatching the signals between the heart and the sutures or vice versa?
Vice versa. That's a good question. Let's see.
Not
For
yet tested, right?
now you have been tested, sir.
Okay.
Yeah.
So this is fixed rate stimulation, that's correct?
Yes, sir. Fixed rate.
And because you know that they're on demand or by standby pacemakers, yeah?
Yes, sir.
That match it, yeah? And you also mentioned, in reference to other question, yeah? The amplitude, what is the duration of the pulse that you generate?
The duration?
Of the pulse?
Oh, not too long.
The hertz.
Yeah.
The hertz?
No. Frequency, it's another story.
Yeah. The
Duration of the pulse, yes? And frequency, it's basically you have presume frequency of the cardiac beat, yes? This is 60 or 70, yeah?
Yes, sir.
Yes.
Do you have
Each one,
references
yeah.
to that?
But duration of the pulse,
Duration?
Yeah.
I'd like to say in milliseconds.
magic,
So not that long.
Yeah, but it was said that it's not a fixed rate, it's actually a conductive rate to the
Yes,
other
it can
cells.
vary.
So it's not a fixed rate.
No, we stimulate to these points, and then from there we try to see what kind of conditions it will be be being at most optimal rate.
I don't know, I was always amazed that like your beat rate of the heart is not controlled by your brain.
No, it's not. It's mainly
It's very
controlled.
interesting, it's really not controlled by
yeah,
brain.
like the pacemaker, they have hysteresis, yes? Basically they stand by and switch on it below the certain threshold and stop about certain, yeah, but that's a different application.
That's why our goal is only to be a temporary pacemaker, not to be a permanent pacemaker. Only to deliver the frequency when it is needed.
The next one. Yeah.
Yes, sir.
So, how are you powering the pacemaker?
pacemaker to this Raspberry Pi. It's got a micro USB outlet connected to this. To
So
it.
the Raspberry Pi should be connected to the human at all times when it is installed inside. Right?
Oh, no, that's actually just, this is only for research purposes. That would be future direction. For that would be a completely different design setting.
Okay. So it's like you're taking the power from the heart or like from other organs?
It would be self-generated, most likely, possibly through quartz. Piezoelectricity, only because it's non-invasive and it'd be constant low stimulation. Most likely we'll be using different types of resistors in order to control that sort of outflow.
So,
Yes, sir.
what, it's, as I expect, like, it's a memory gel suture that
Yes.
will beat, right?
Yes, a hydrogel.
And does it have any lifespan to be? Does it reach to a point where it stops,
After a while.
or you have to keep on updating, or?
We will most likely have to refresh our cells, most likely because each cell line, they only last a certain amount of time before they either over-proliferate and they have too many cells on the suture. So from that would be one of our main tasks, which is to see how long these will last in future direction.
We culture them roughly 4 months.
4 months?
Yeah.
That's a decent time.
It is a decent time.
And so Melvin started one last summer, and we had to close it down at Christmas because we're not there at Christmas.
Question?
Yeah, so have you listed using like an actual digital to analog converter instead of sending PWM to the part? I mean, like would the cells respond differently, like with a real analog voltage rather than like a modulated signal?
They would respond differently only because with modulation, it would be more direct compared to what we have right now.
Right. Okay. And is there any reason why you cannot use like an off-the-shelf pacemaker for that? I mean, I'm guessing right now, like in the research stages, you would want to use that, but then like in the patient, would you
For
use?
patients, that is a tried and true method. That would be most optimal. From what we have is more student-based where anybody can jump in. It's a friendly user interface, and from there we can see how our cells would be able to react in different frequencies for data input. We'd be able to analyze the humidity, we'll be able to analyze what are the best optimal ranges for our cells, and from there we're able to publish, and other people can reproduce our results.
Also, I might add, a commercial pacemaker is cheap at $3 grand.
Yeah, so this is
So-
only 30 bucks.
Yeah, that's why we're where we are.
Any other questions? Anyway, thank you very much. We have to move on now.